back to me with something so I can look at. The article is related to biofilms. So what I would suggest you do to diversify the articles is maybe focus on a couple of the growth proteins that are out there. The MREB protein, the FIT-Z protein. So do a search on that, see what types of research have been done on those, because that introduces exactly what we've been talking about so far, right? Those two proteins play a really important role in the segregation and in the septation process, right? So the question is, what types of research have people been doing on those proteins to give us a sense of where that might take us in the future? Okay, so I think that would be an excellent article. So be thinking about that because the biofilm topic's already been taken. Okay, so that's going to happen on Monday. So we'll dedicate all our time for that. And there will be one question related to the presentations on the exam the following Friday. So we've got presentations on Monday. We have an exam review on Wednesday, and then we have our first exam on Friday. Okay. So it's really important that you be here for the presentations because the questions that I'll create will be based upon what goes on in that presentation. Okay. So make sure you are present as well.
You said the structure of the exams like what we just did.
Exactly. It's all short answer, essay, if you will. That's basically it. Yeah. So you have to be prepared to know your stuff well, be able to convey that to me in a clear fashion. So that's important. Okay, so that's the game plan. So we're slowly coming to the end of our topic for our first exam. So make sure you are on top of it. This weekend should be busy for you to make sure you got everything together for that exam next week. Okay. All right. So let me introduce this last topic. This topic really focuses on cell structure. Okay. Again, we're focusing on bacteria. So when we talk about bacteria, we know that in terms of structurally, there are cells within the bacterial community called gram-positive and gram-negative cells, right? And that's essentially what we see up here. And so the big difference between the two are various aspects of the structure that sort of stand out. Number one is this structure known as peptidoglycan, and that's what we're going to really focus on today. But if you kind of look at the overall envelope of these cells, gram-positives have what we call, books refer to it as very thick peptidoglycan layer. And that inner membrane, of course, maintains the cytoplasm. Whereas gram-negative, you still have peptidoglycan, but it's a much thinner version of that. And you have two membranes that we call the inner membrane, which is similar to that, and then you have this outer membrane as well. That's quite different for gram-negatives. So, and I just included this morning, but you can find it in chapter 3. So this is a cartoon image of the two as well. Okay, just to give you an idea. So the complexity of the cell wall is quite complex. Even though this looks complicated, this is only scratching the surface in terms of what else is present and why that's so important. And what we're going to do is just touch a little bit with some detail about one of these aspects of the cell wall, and that's the peptidoglycan. If you're really interested in this, I teach a course called Bacterial Cell Walls. It's a whole semester course, and we focus on some really interesting things about these structures that are really important for not only cell physiology, but in terms of pathogen-host interaction. You know, how our bodies recognize these cell walls. Because what we discovered is that a lot of these components are really important for why we get sick or why we don't get sick. So it's really quite interesting. So as we go down that road, let's think about peptidoglycan and think about what's important. A couple things to keep in mind. Why is it important? Well, peptidoglycan, number one, is a unique structure for bacteria. And what I mean by that is that we don't have the structure known as peptidoglycan. Okay? And because of that, the uniqueness really is based upon the enzymes involved in assembling this unique structure. So if the structure is unique, then usually the enzymes are unique as well. And that's certainly the case. So bacteria have unique enzymes that produce this unique structure. We don't have those enzymes, and we don't have those structures. Okay, so there's immediate division of separation, which is actually ideal because when you think about it, when one has to prevent bacteria from growing, one of the strategies is to be able to use a drug that targets something unique, right? So as you administer drugs to yourself to combat a bacterial infection, you want the drugs to selectively go after a unique structure, not your own structures, right? And that's exactly why antibiotics, particularly the one we'll talk about today, that targets peptidoglycan, that's why it's so effective. It doesn't attack our cells because we don't have that structure, but it certainly attacks bacteria because it does. It's a wonderful antimicrobial. Okay? And then the second thing, just for information purposes, is really thinking about it in terms of the actual structure of peptidoglycan. What we discovered within the last few decades is that it actually is a critical molecule for stimulating our immune system. Okay, we've all heard how bacteria is important for our immune system, right? So what do you mean by that? Okay, the details are probably unknown, right? Well, one of the detailed aspects of how our immune system is stimulated is because our immune system recognizes peptidoglycan structure. And so that's important to know because by knowing that, it's really important for us to then begin to understand how pathogen-host interaction occurs and what we can do to combat that interaction in terms of disease-causing organisms. Okay, so I want to look at a few things in terms of terms that are really important in terms of the actual structure first, and then we'll look at the synthesis today. And then on Friday, we'll look at the actual antimicrobial mechanisms of action and how the resistance develops on Friday. Okay, so today is the structure, getting used to some terminology, and the processes of synthesis. Okay, so the critical steps. So we'll cover those this morning. Okay, so let's take a look at the structure itself. Okay, so if you take a look at peptidoglycan, it's basically a series of sugars. And really, it's two sugars. So these represent sugars. And so those sugars are derived from your basic backbone pathway called glycolysis. Okay. So if you're thinking about where those sugars come from, they come from the precursors of glycolysis. Okay. So as your bile pathways produce these sugars, and I want you to know the names of these sugars rather than just the abbreviations. Okay. So we abbreviate this a lot of times as NAM, and we abbreviate this in many cases as NAG, right? But I do want you to know the names. So that's the first thing that you really need to understand. I need to know the name N-acetylmuramic acid and N-acetylglucosamine. They're very similar sugars. And I also want you to know that they're a 6-carbon sugar. Okay? And so the 6-carbon sugar is important because it defines this chemical structure. Okay? So I do want you to know that So if you take a look at that structure here, second thing I want you to know is number one is the 6-carbon sugar. And the labeling of the sugar defines how some of the terms are used to describe how the links occur. Okay? So in other words, you've probably heard how these sugars, when they're linked to form the peptidoglycan, the important link is called the beta-1,4 linkage. Okay? If you haven't heard that, what's the beta-1,4 linkage? And I want you to understand what that means. Well, beta has to do with the position in terms of its orientation of this oxygen molecule right here. Whether it's above the plane of the sugar or below. So you're going to have beta or you're going to have alpha, up or down. Okay, so the position of that is important. Second one is the numbers. What's beta, what's 1 and 4? Or that's the carbon number, right? So if you look at the 6-carbon molecule, what you do is you look at this oxygen molecule here, and you work your way to the left clockwise, and it goes 1, 2, 3, 4, right? So you can see how that works. 1, 2, 3, 4. Okay. So now you've got carbon number 1 associated with carbon number 4. Hence the name beta 1,4 linkage. Okay. So that's a covalent bond, and that bond happens because there's a specific enzyme called the transglycosylase. Okay, so there's a protein that serves as an enzyme that creates this bond. And when that bond happens, what we call these series of sugars, your book calls it a saccharose, but I also want you to know this term, call it glycan. G-L-Y-C-A-N. Okay, that's just another term we use to describe the alternating sugars, NAM and NAG, as it's undergoing its building process. Okay. The second part of the peptidoglycan that's important would be there's a bridge, a really important organic molecule that bridges the sugar to a series of amino acids. Okay, and that's this molecule right here. This is what we call the lactyl group. Okay, and that lactyl group is associated with the NAM sugar, and that's part of why it's unique. Okay, and so it bridges and it allows the attachment of this 5 amino acid polypeptide. We call that the stem peptide. Okay, so the basic structure of peptidoglycan is 2 sugars and the stem peptide connected via the lactyl group. Okay. Now, the sequence of this peptide is typically in a series of 5 amino acids. So we call it a pentapeptide. Okay. In general, all bacteria will produce 5 really unique amino acids. And I say unique in that what we mean by uniqueness is that amino acids, like sugars, you think about your days of chemistry, orientation is everything in terms of how enzymes recognize molecules. Okay, and if you're following me, you'll understand this. But let me get a little bit more clarity in terms of what I mean by that. Now, enzymes recognize molecules that are really unique based on their orientation. Okay, now, remember your chemistry days. When you think about a molecule, one of the ways in which we understand molecules is based on their orientation in terms of what we call the L and the D forms, right? And that's just basically the mirror image of molecules. Okay, the best way to understand that, if you remember, is what we call an isoform, is your hand. Okay, my hand is essentially the same. Okay? And when you match them up together, that defines how they're very similar. But in essence, they're very different. Okay? Because if you really think about it, this side of my hand versus this side of my hand, if you were to face it toward you being the enzyme, it's completely different. So what happens is enzymes recognize a unique form. Let's say this is the L form, which is the most common form of molecule that enzymes recognize, the L form. If you have a D form, which is different, right? It's still the same molecule, so it can be the same sugar, but it's a D sugar. Now your enzyme doesn't recognize that because my thumb is the other way, pointing the other way. So nature is really interesting because they don't recognize that. So you can actually feed a cell, and cells prefer L forms of sugars. You can give it a D form sugar and it won't grow, absolutely won't grow. You're like, wow, this is because of the forms are different and the enzymes don't recognize it. It's the same with other molecules like amino acids. So if you notice that these amino acids have these L and D forms of amino acids. So in nature there are also L-alanines and there are D-alanines. And so bacteria utilizes the types of forms that are less present in our cells. So our cells prefer the L, but bacteria tend to use these D forms. And so now it begins to make sense, because if it's a D form and enzymes recognize it, then those enzymes must be unique and different. Okay? And that's where the differences happen. Okay? And so they tend to have these unique D forms. And the last thing I want to say about structure is that the series of 5 amino acids, as they're attached to the NAM sugar, the last two, in general, fairly conserved in terms of how they're displayed, but it can differ based on how they respond. But the last two are typically 2 alanines, and that's going to be important for the later lectures. Okay, so the last 2 amino acids. So if you label these, you usually give them numbers: 1, 2, 3, 4, and 5. So 4 and 5 amino acid on the end of that stem peptide are typically always 2 alanines, D forms. Okay. And so during the synthesis process, that plays an important role. Okay. So just to review, 2 sugars connected to a series of amino acids called the stem peptide. And the end of the stem peptide has this unique D form alanines, and that plays a really important role in synthesis. Okay. So now we're just looking at this process. Okay. So now, as far as synthesis is concerned, there's going to be an important process that happens. This kind of sums everything up. Okay, so how does it all begin? So I'll talk about a few details that I think are important to understand synthesis. Number one, as I said, the 2 sugars come from the backbone of glycolysis. Okay, so as they form these sugars, NAM and NAG, the question is which one comes before the other, right? Because what happens is generally, in terms of biochemistry, the pathways usually come in a series of events, right? So within this process of peptidoglycans, if you orient yourself, the top part is the cytoplasm, okay, and then the bottom part is this is the cytoplasm, and then it's going to the top is the cytoplasm, and it's going to flip across this way. So let's look at the details. So the top part's your cytoplasm of the cell. That first molecule that I want you to be familiar with is called the UDP. What's the UDP? That stands for uridine diphosphate. Okay, another important organic molecule comes from the backbone of glycolysis, right? And so what that is it's called a carrier. So make sure you define a UDP as a carrier. So what's a carrier? Well, if you look at the images up here, it's attached to the NAM. So it's attached to that sugar. And so if you thought about how the assembly happens, it actually depends upon this UDP carrier to be successful to assemble the 2 sugars together. So what happens is you get the attachment of the carrier in the cytoplasm, which then, remember, the NAM is what has the stem peptide. So during that process of assembly, the NAM begins to accept the individual amino acids. And there's an important point I want to make about that. Okay? So let's compare that formation of a polypeptide, which is a series of amino acids, to another event in the cytoplasm called translation, in which it's essentially formation of a series of amino acids as well, right? But we call that protein synthesis. Now, in protein synthesis, as you're attaching one amino acid to another, that requires the messenger RNA, requires the ribosome, and requires the transfer RNA to deliver the amino acids individually, right? And it forms this long amino acid, and that becomes a protein, right? So if you think about it, when amino acids come together to form a polypeptide, the process in which this polypeptide forms is completely different. Okay, so number one, it doesn't rely on a ribosome. Okay, so how does that happen? Well, what happens is individual amino acids that specifically recognize the lacteal and the first amino acid, which is the L-alanine, is recognized by that enzyme, and it forms that first peptide bond. So we can call that enzyme 1. And then another enzyme recognizes glucose, D-glucose, and then it forms that peptide bond. And a third enzyme called enzyme 3 recognizes that third amino acid called L-lysis or DAP, so on and so forth. So now you can begin to understand that this assembly of this polypeptide doesn't require a messenger RNA, it doesn't require a ribosome, but the similarities of this assembly, it does rely on a transfer RNA. Okay, so the delivery of these amino acids, it doesn't show it in this image. So the amino acids aren't just floating in the cytoplasm, they're actually attached to a ribosomal RNA. And so that's delivering these, and the enzymes are recognizing them and attaching them. And so when this whole process is done, you have a series of 5 amino acids attached to the first sugar. Okay? So that's really, really interesting because now, as far as microbiologists, if we're looking for a target, because one of the best ways to target bacteria would be to develop a drug that binds to an enzyme that's unique. Okay, I've already mentioned possibly 5 unique enzymes, right? That could be involved in this. So really, they're actually quite wonderful as we discover more details about how this process happens. So when that happens, then what goes on is then this carrier brings that molecule, pentapeptide, close to the membrane, cytoplasmic membrane of the cell. And what it does, it then associates with another molecule that's found internally. I want you to know this, that molecule is called the bactoprenol. Okay, so there's a bactoprenol, and that bactoprenol resides within the membrane. And what's unique about that is, why does it reside within a bilipid membrane? Well, a couple things, if you think about bilipid membranes, is that lipids are very hydrophobic internally. They don't like water. So the heads are hydrophilic, so they stick out and they interact with water. So if you ever wondered how a lipid forms this bilayer, what happens is that the hydrophobic regions attract to each other in an area where there's no water, and the polar ends, heads stick outward, they interact with water. So if you can do these experiments actually in the lab quite well. You can take individual lipids, put it in a solution, and those lipid heads would immediately stick outward, interact with water, and the hydrophobic tail would go internally where there's no water. It would assemble, form this nice circular, what we call a mycel. And nature does that really well with these guys. So anyway, molecules that are hydrophobic, like a bactoprenol, prefer to be inside there. And so that's where they exist. And so they're there, we think, for a reason, because they then interact with this NAM carrier attached to it and basically transport it across the membrane at some point during the assembly process. Okay? So it attaches to it, and before it actually transports itself, that assembly has to undergo further synthesis. So that first portion of the structure where you've got the NAM, the pentapeptide attached to the bactoprenol, we call that the lipid 1 synthesis. Now, I always, dislike that term because lipid 1 always suggests maybe it has something to do with lipid synthesis. It has nothing to do with lipid synthesis. It's just a stage in synthesis that's defined as the lipid 1. Okay, what that means is you've got one of the sugars that has the pentide and it's attached to the bactoprenol. Okay, now as the assembly goes forward, that sugar now. Oh, question.
Just backtracking real quick. Did you say that the peptides were brought by ribosomes?
No, they're not. They're not. And that's what makes them different. Two things are different. No ribosomes, no messenger RNA. Only enzymes that attach them individually. What's similar is the transfer RNA. Okay? And it's really interesting how this decision happens in bacteria because you have a messenger or transfer RNA attached to amino acids. It's normal. And it has to go through a decision process. Do I support translation or do I support pentapeptide peptide-like information? And there's a need for that. So the supply of transfer RNA is really important. If that supply is low, it can affect cells and how they can survive in the future.
So bullet point number 2, what does it mean by saying HP is used to feel this, but tRNA and ribosomes are not involved?
What they mean by that is that the transfer RNA and its association with the ribosome in translation. Okay, and I know that image is basically what that means is the transport RNA still attaches to the amino acid in the cytoplasm. That's still there, okay? And so as it's there, then it doesn't associate with the ribosome, but it associates independently to deliver the amino acid to this formation of the peptide. Okay? So then we would call that the stem peptide synthesis. If the transfer RNA came to a ribosome and delivered it there, then it would be translation, right? So as I said, there's this fork in the road. That there's really some interesting studies that talk about that concentration of transfer RNAs and which direction they go in terms of supporting cell survival. It's really quite interesting. But what's interesting, and the point I want to make, is that's the conservation part of it. So if I ask you the question, what's conservative about this process? What's conservative is that stem peptide formation and translation both need the transfer RNA. What's different about it is that stem peptides do not require ribosome, do not require messenger RNA. Translation does require ribosome and does require messenger RNA. The outcome's the same. You're building a polypeptide, a series of amino acids. Okay. Of course, translation is a much longer polypeptide because it's a protein, but this one's a series of 5, but nevertheless, it's the exact same thing. The N-terminal part of the peptide bonds to the C-terminal part of another peptide. Absolutely the same at that point. It's just only 5 amino acids long. Right? And that's as far as it goes. So that's the point I want to make. Make sure you can kind of see how that is. Okay? So you go through this transformation, lipid 1 to lipid 2. So what's the difference? Well, the only difference between that is now you've attached the second sugar, okay, which is the one that doesn't have the pentapeptide, and that's the NAG sugar. So now you've got 2 sugars. One of them has the stem peptide, and we call that the lipid 2 stage. Okay, and it's still attached to this bactoprenol. And at this point now, it's really interesting. How does that molecule get from the cytoplasm to the periplasm? Because that's where the peptidoglycan is, right? The bactoprenol is the key molecule that allows that molecule to, there's a lot of terms you use, flip, transport. It basically takes that lipid 2 structure, flips it across, and now puts it on this side. Okay, the bactoprenol does that, so you've got to have that. That's really key. So once now it's on this side, now this is where the peptidoglycan exists. So now think about that growth process, particularly septation, right? Remember when cells segregate and the septation forms, the cells begin to pinch inward? I mentioned that right in there, you've got a tremendous high rate of peptidoglycan synthesis occurring. So if you can imagine, all this right here is happening right at that septation stage where that process is happening. Okay? Very highly active in terms of that process. Okay? So those are the key pieces that I want you to begin to understand in terms of the synthesis and the structure of peptidoglycan. Okay? So we'll continue this lesson on Friday. We'll transition into the microbial part and the resistant part as well. Okay? All right.
Yeah, I was thinking.